And also we can think about the risk of formation of reactive or toxic metabolites. So we know that metabolism by P450s can lead to the formation of these metabolites. We might observe experimentally, for example, time-dependent inhibition, which is caused by irreversible binding to the P450 enzyme. Or, for example, following metabolic activation, you might see formation of glutathione conjugates. Now, to be clear, we cannot yet directly predict the reactivity of metabolites. I'd say watch this space. It's some active research that we're working on. However, what we can do is look for patterns of metabolism that correlate with the formation of reactive metabolites. In this particular example, we'll look at a series of PDE-10A inhibitors published by Chino et al. A few years ago. And this is an example of one of the compounds from the series. You can see here that in the paper that they suggested that the metabolism at the 7 position on the imidazopyridinone ring may be responsible for formation of that reactive metabolite. But what's interesting, if we actually look at the predictions for this compound and a number of others within that series, here you can see the predictions is really that the vast majority of metabolism is likely to occur due to O-demethylation or N-demethylation here. And in fact, there's no predicted metabolite, no significant predicted metabolism at that 7 position. On the other hand, what we can see here is a strong correlation between N-dealkylation on that same ring system here, and the experimentally observed time-dependent inhibition. This is shown here as the percentage activity remaining after a 30-minute incubation. And so a low value here indicates a high degree of time-dependent inhibition. And so this correlation, admittedly, it's only 3 data points here, but there's a very strong correlation between the degree of N-demethylation and this time-dependent inhibition, which would suggest that reducing N-dealkylation is one approach to improving the risk profile of these compounds. It's likely that the N-demethylation there leads to rearrangement and activation of that ring system.